Thanks, Elaine. So I will talk more about CAR T cells. Disclosures are here, primarily with Novartis and Immunotherapies. So cancer immunotherapy is not a new idea. And Lori Glimcher spoke some of this. It's over 100 years long. It's just our tools have gotten much better. And many of the things that were tried a long time ago that failed, we now understand why with various, because of the new technologies we have. The two that have really changed the face of cancer therapy now are the checkpoint therapies, which received Nobel Prize on October 1st, and CAR T cells. And until then, there really had been no FDA approval, and so just really changing things. So CAR T cells are complementary to checkpoint therapies. Checkpoint therapies augment the natural immune system, and CAR T cells are a form of synthetic biology. So using gene transfer technology, cells are created that don't exist in nature. And so they're a synthetic polypeptide introduced generally with viral vectors. In our case, we use HIV or a lentiviral vector. The two that are marketed are similar in many ways, but also have some technical differences. Gilead markets Yescarta, and Novartis, Kymriah. They have different signaling domains inside the cell, but they have exactly the same binder that binds to the molecule on the leukemia cell, which is CD19. So it's an antibody fragment. So CAR T cell therapy is really quite different than other drugs. It's really a process and more akin to bone marrow transplantation. And that creates some of the issues we'll talk about on pricing. So it begins with the patient donating their own cells in the blood bank, then a manufacturing process that takes about 10 days, where the cells are in a laboratory activated and transduced with a viral vector to express the CAR molecule in the T cells. Cells are then frozen and returned to the hospital, and then they're in infusion-ready DMSO. They're thawed at the patient's bedside and infused in a 5-minute infusion. At this point, they're all autologous, the patient's own cells, but there is a lot of research underway where we could have so-called off-the-shelf cells, which could be derived from cord blood, peripheral blood, or even induced pluripotent stem cells. And that would really change the manufacturing approaches if that could happen. This is the story behind acute lymphoblastic leukemia, which occurs in about 5,000 cases a year in the US, but only about 600 pediatric cases relapse due to the effectiveness of high-dose chemotherapy and radiation. The approval that happened last August for leukemia age 3 to 25 is for second or more relapse in young adult or pediatric leukemia. The first adult patient that we treated, you saw the first pediatric patient, but this is an example of the first adult patient. He had, by genetics, a hypodiploid ALL, which never responds to chemotherapy. He's a physician scientist. He's come out into disclosed as HIPAA. It's Keith Eaton, a professor at the University of Washington. And we treated him, and he had a very high on-target toxicity, as Emily did when they said he had a 107-degree fever that lasted 3 days, and then he's completely responded. He's back full-time in a lab. He's an MD, PhD. At the bottom, he wrote a summary of the cost that it cost him. So he was on our experimental protocol and cost nothing, but the rest of his ancillary, such as ICU stay and so on, ran up to $500,000. So that's an issue we need to discuss. So those are approvals now for CD19 CARs. It's in lymphoma and leukemia. Next year, we think that it's likely 3 different companies will get approval for myeloma, of which there are 30,000 cases a year. Right now, myeloma is probably the worst cancer as far as financial toxicity because it's only 2% of all cancer, but 7% of all costs due to the fact that patients live a long time but they don't have a definitive therapy. The major scientific challenge in the field is, can this work in solid tumors? I think we'll have approvals in nearly all blood-derived heme malignancies of various kinds of CAR therapies and other approaches being tested. In solid tumors, there have been no home runs yet, but there are a number of different kinds of CARs being developed in the various tumors that are listed here. And there are also T cell receptor expressing cells. So synthetic biology using a T cell receptor rather than a CAR molecule. And then the other issue really is how can we make manufacturing more efficient and economical? This is really an engineering issue at this point. You know, I mentioned there are patient-specific cells. It's done individually on every patient. There are issues of limited blood banking apheresis right now. The GMP suites where these are made, the labor to manufacture the cells is limited. We have an issue called peak serum, which is related to the concept in the oil, where we're going to run out of oil and have a peak oil supply. So right now, the CAR T cells are grown in human AB serum, and there's only so many donors that can supply serum. So we really desperately need to have serum-free media, and that's listed there. Another issues related are the geographic disparities and high cost of these cells. So on the left side is a heat map of where CAR T cell trials, there's over 300 of them now in the world, and the most common place where they are is China. Second is the US, but if you notice, the Southern Hemisphere is completely devoid of trials, and as well as Europe is relatively devoid. They're not very adoptive of new technologies involving gene modification. The Wall Street Journal had an article last month on the $1 million cancer treatment, who will pay. So in the case of myeloma, it was talked about there are 30,000 new cases a year in 2017. And at the cost of current standard of care, the total lifetime cost to treat all of those patients will be $22 billion. This is clearly not sustainable. We need curative therapies that are affordable. So this is what I'll leave you with, where we're at. Right now we have new to our armamentarium in cancer therapy, engineered cell-based therapies and antibodies such as checkpoint therapies. None of these will be given alone, except maybe in the unusual case of leukemia. I think what you've heard throughout today is the theme will be there'll be combinatorial combination therapies, which adds then tremendously to the cost. And with that,